episode of Myeloma Crowd Radio, a show that connects patients with myeloma researchers. I'm your host, Jenny Ahlstrom. This is our 102nd show, and I'd like to thank our episode sponsor, Takeda Oncology, for their support of Myeloma Crowd Radio. Now, I hope you all enjoyed the recent eclipse, because we did. The Myeloma Crowd hosted an eclipse camp in Rexburg, Idaho, and we raised over $11,000 for the Myeloma Crowd Research Initiative. This means we've now raised $488,000 towards our $500,000 goal, and we are only $12,000 away from meeting that goal. As you hopefully know by now, the Myeloma Crowd Research Initiative is funding 2 immunotherapy projects. One is a CAR T-cell research project at the University of Würzburg in Germany, targeting CS1 and BCMA, and the second is called MILS, or marrow-infiltrating lymphocytes, which is an immunotherapy used with stem cell transplant from Johns Hopkins. Now, today's show is a fascinating one, which I ran across several months ago. It was an observation that sugar molecules can essentially act as a smokescreen for myeloma cells. And when we posted an article on this, Dr. Michael O'Dwyer from Ireland retweeted the article on Twitter and noted how key this is for myeloma. I had the wonderful opportunity to meet him in person at the European Hematology meeting in Spain in June and invited him to participate on the show. So welcome, Dr. O'Dwyer.
Thanks very much, Jenny. I'm delighted to be on the show.
Well, it was so fun to meet you in person, and it's wonderful to have you here. So let me just give a brief intro for you, and then we'll get started with some of our questions. Dr. Michael O'Dwyer is director and founder of the Science Foundation Ireland and Irish Cancer Society Blood Cancer Network, and chair of the Hematology Lymphoma Subgroup of All the Ireland Cooperative Oncology Research Group. He established the Multiple Myeloma Translational Research Program at the National University of Ireland, Galway, and was the first to run phase 1 clinical trials for blood cancers. Dr. O'Dwyer is a professor of hematology at the NUI Galway and HealthTree and Health Research Board clinician scientist there as well. He's a visiting scientist at Dana-Farber and the Harvard Medical School. Dr. O'Dwyer has 6 patent applications, including a particular marker that you can say and talk about today for multiple myeloma, and patents involving natural killer cells. He is co-founder of a company called Oncocellular and Oncommune, 2 startup companies focusing on NK cells as anti-cancer therapy. Oncommune's research is evaluating how NK cells can be optimized for use in myeloma therapy. Dr. O'Dwyer, interestingly, treated the first patient worldwide with an E-selectin inhibitor in AML, which is leukemia, which received breakthrough designation from the FDA, and he'll be sharing a little bit more about that in myeloma. And he's now running the first clinical trial for E-selectin inhibition in multiple myeloma. So welcome, Dr. O'Dwyer. We're just thrilled to have you. We know you have extensive experience in immunotherapies and are looking forward to understanding these sugar molecules a little bit better.
Thanks, Jenny.
So maybe we just want to start. When I was doing a little bit of reading, it showed up as being called sialic acid sugars. So maybe you can just start by sialic.
Sure. So it's sialic acid. So I guess if we buy something to eat or whatever, and we look at what's on the label, and it tells us that there's carbohydrates, proteins and fats. Well, basically, our body is made up of these different building blocks, along with essential minerals and so on. But carbohydrates is, I suppose, a term that encompasses all types of sugars, and some of our sugars are for energy, like glucose and so on, and glycogen is a storage form of sugar. But there are other sugars that actually have a different function. So sialic acid is a non-energy type of sugar, and it's normally expressed on the surface of many cells in the body. And on the surface of cells, it decorates proteins and lipids or fats on the cell surface. And the addition of in fact, different types of sugars to proteins or lipids is a process that we call glycosylation. And it's, sometimes referred to as one form of post-translational modification, which means that when your genetic code is made into mRNA, which is then made into protein, that this is a way of further modifying and making even more complex these different proteins. So the sugar coating of proteins and lipids on the surface of cells can greatly influence the function and so on of these proteins. And an example would be, for example, erythropoietin or EPO, which some patients with myeloma may sometimes receive to stimulate blood cell production. This protein or growth factor needs to be sugar coated for it to work properly. The same thing with monoclonal antibodies, daratumumab and elotuzumab are example of monoclonal antibodies. These are frequently glycosylated or sugar coated as well. So basically sugars attached to proteins is actually a very, very common thing. Now, if you were to actually look down with a very, very powerful microscope, such as an electron microscope, at the surface of cells, what you would see is that they have a kind of a fuzzy outer halo on the outside of the cells, and this is actually composed of sugars. So when one cell encounters another cell in the body, the first thing that they sense, in fact, is the sugar coating on the outside of the other cell. So the sugar composition on the outside of the cell is very, very important in how cells interact with one another. And of the different types of sugars that are involved in, if you like, cell-cell communication or how cells adhere or stick to one another, sialic acid is probably one of the most important sugars. Now, in terms of the binding then, or the interaction of proteins that are on one cell interacting with sialic acid, proteins that bind to specific type of sugars are referred to as lectins. And this terminology is, I hope it's not sort of too complicated, but there are two different type of proteins or lectins that bind to sialic acid. And this is relevant for our further discussion because these two main proteins that bind sialic acid are referred to as the siglecs and the selectins. And a lot of my research in this area is based around understanding how sialic acid on, in this case, myeloma cells interacts with siglecs expressed on immune cells or selectins that are expressed on blood vessels.
Mm-hmm. Okay. And then what's the relationship with the sialic acid and these siglecs and selectins with the tumor cells?
Yeah. So then if we take the siglecs first. So the siglec is a kind of the name is a shortened version of sialic acid binding immunoglobulin like lectins. So they're proteins on the surface of blood cells, especially immune cells, that have a sort of a structure that's similar to immunoglobulins or antibodies, and these siglecs selectively bind sialic acid and particular types of structures that are coated with sialic acid. And the expression of the siglecs appears to be particularly important on the surface of immune cells such as macrophages and natural killer cells. And we believe that they play an important role in regulating the immune responsiveness of these cells. And in most cases, the binding to sialic acid stimulates the siglec, which dampens down the immune response, because siglecs they're a little bit analogous or similar to PD-1 receptors. So I think many people are probably familiar with the checkpoint receptor PD-1 that can be expressed on T cells and in fact NK cells as well. So if you think of sialic acid on, for example, a tumor cell, which could be a myeloma cell, and Siglex on an NK cell, in a way it's a little bit analogous to PD ligands on a tumor cell interacting with a PD-1 checkpoint receptor on an immune cell. Therefore, I think we can consider that Siglex are somewhat like checkpoint receptors on NK cells, and they also seem to play a similar function on macrophages.
So you have the sialic acid on the myeloma cell, and you have this Siglex on the immune system cell, and what you're saying is they're talking to each other in a certain way depending on how much of each are on each type of cell?
Exactly. Now, an important thing, is that, sialic acid is expressed on the surface of many different cell types, but what is important is that in cancer, that this expression of sialic acid sugars seems to be exaggerated, and why this is the case. We've done a little bit of work, looking at this in myeloma. We have found that myeloma tends to have a very high expression of sialic acid, and it doesn't appear to be due to any mutations. It doesn't appear to be due to increased numbers or copies of the genes. It appears to be more related to increased expression of the genes. And what we've actually found is that some of the factors within the microenvironment, in the tumor microenvironment, that we know aren't a good thing in myeloma, such as cytokines or things like interleukin-6, IGF or insulin-like growth factor, and low levels of oxygen or hypoxia, all things that we know are bad in myeloma that drive more aggressive disease. All of these things are associated with stimulation of the genes that are code for the enzymes that are involved in generating sialic acid on the surface, and these enzymes are referred to as sialyl transferases.
So is this true for everybody? So what you're saying is, regardless of the genetic features that you have for your type of myeloma, that you find that everyone or just some people have this increased level of sialic acid?
No,
I
think
these sugars on their myeloma cells.
I think that what we find is a variable level of expression, and in some cases it does appear to be much higher than in others, and kind of our own sort of meeting was a little bit serendipitous in a way, but in fact how I got into this area in the very beginning was really quite serendipitous as well, because I'm a hematologist. In fact, I started my career initially in CML, in chronic myeloid leukemia. I worked as a fellow and subsequently as a junior faculty on the development of imatinib, which was the first kinase inhibitor for CML, with my mentor Brian Drucker in Oregon, back in the early sort of 2000s. But when I was resurrecting my academic career in Galway, I had a sort of a chance encounter with a glycobiologist. Now, glycobiology is the study of the biology of sugars. And over a cup of coffee, we were kind of sharing experiences with each other, and what our respective areas of interest were in terms of, in his case, glycobiology, in my case, blood cancers and leukemia and myeloma. And when he started explaining to me what sugars did, immediately I thought, it's very likely that they must play a role in what we call adhesion-mediated drug resistance. So when, cancer cells stick down on a surface, they often, in the bone marrow or on blood vessels, they often become more resistant to chemotherapy. So that's what sort of stirred my interest originally, and my colleague Lokesh Joshi and I decided that we'd put together a project, and we had a very bright young hematologist called Siobhan Glavy, who subsequently went to work with Irene Gabriel at the Dana-Farber, and in collaboration with Irene Gabriel and Gareth Morgan, we had access to sort of more resources than we would have had here in Galway. And what we found was that a number of different sugar-related genes were important, but the one that caught our eye as being important in terms of its impact on survival, when we looked at Gareth Morgan's UK data from one of the big myeloma trials that they did in the UK, was this gene called ST3-Gal6. So it is a sialyltransferase, so one of these enzymes that puts sialic acid on the surface of cells. Now, what we found was that there was a range of expression of this enzyme in patients, but if you looked at those patients who had the higher levels of expression, that they had a much worse overall survival and a shorter time to progression on treatment. And when we looked at the time at other factors that could be responsible for poor prognosis, such as genetic factors and stage of disease and everything, the expression level of this particular gene, this sialyltransferase, was in fact independent of those other recognized risk factors. So we thought that this was quite important, quite novel, because it was the first time really that something like this had been implicated in multiple myeloma.
That's so fascinating. So what you're saying is the more of this expression that you see of that particular gene, or then the lower the immune response, or the more drug resistant it tends to be?
I feel I'm on steadier ground when I talk about the drug resistance, because our research is at a more advanced stage than our work on the immune side of it. But we have growing data supporting the fact that increased sialylation or expression of sialic acid on the myeloma cells does impair the function of immune cells. We have strong data in the laboratory to support that. But we need to extend that, I think, a bit further and show in animal models, for example, that is definitely the case. But we can take parallels from other cancers, and some very nice work has been done in other settings, such as in kidney cancer or in breast cancer, showing that the expression of sialic acid on the surface of tumor cells is associated with reduced responsiveness of natural killer cells, for example. And Caroline Bertozzi from Stanford had a very nice paper in the reputable journal PNAS back in September, I believe, where she showed a very interesting approach where they used a monoclonal antibody, Herceptin, or trastuzumab, which targets the HER2/neu antigen in breast cancer. And with the antibody, what they did was they conjugated or added to the antibody an enzyme known as sialidase. Now, this is an enzyme that actually removes sialic acid, cleaves it off or removes it from the surface of the cell. And when the antibody was combined with this enzyme that removed sialic acid from the cell surface, they saw much greater enhancement of killing of breast cancer cells by the HER2 antibodies in the presence of natural killer cells. Now, I should explain, maybe just in case people aren't aware of, one of the main mechanisms that antibodies use, that monoclonal antibodies use to kill tumor cells, is activating natural killer cells. So when an antibody binds to its target on the surface of a tumor cell, the natural killer cells are then able to recognize the antibodies because they have receptors for antibodies on their surface, and they can then bind to the tumor cell via the antibodies and release their toxic payload of granules into the tumor cell that kills the tumor cell. So basically, in the absence of the sialic acid, using this novel antibody, there was very significant enhancement of the killing of these breast cancer cells by natural killer cells. So what we've
kind of
done,
like wiping a clean slate, kind of, so to let the natural killer cells do their job.
Exactly.
Mm-hmm.
So what we have done in my lab is, first of all, we have seen that myeloma cells, initially we just looked at cell lines, but we saw that all the cell lines that we looked at express the types of sialic acid that will bind to Siglecs. So we refer to these as Siglec ligands. So we found that myeloma cells express these quite strongly. Then we decided, well, that's all well and good, but these are only cell lines. So we look at patient samples, and we found that in patient samples that we can also see that these sugars that bind to these Siglecs are also present on the surface of patient samples. But we also looked at natural killer cells from patients, both in the blood and in the bone marrow. And in all the samples that we've looked at, we can see that there's strong expression of Siglecs on the patient's natural killer cells. Now, we then have gone and looked at natural killer cells in the laboratory, and we have shown that when you pre-treat myeloma cells with an inhibitor that removes the sialic acid from the myeloma cell, and that in itself, isn't in any way toxic to the myeloma cell, and combine then, or put the myeloma cells in contact with the natural killer cells in what we call a co-culture, that the natural killer cells are much more efficient at killing the myeloma once this cloak of sugars has been removed from the surface of the myeloma cell. So we think that this is important. An important way for myeloma cells to evade killing by natural killer cells. Of course, you could argue, well, maybe there's some other reason why removing the sugars has made the cells more sensitive to killing. So what we also did is we used a genetic approach to actually knock down the expression or reduce the expression of the Siglec receptors on the natural killer cells, and we saw something similar, that there's enhancement of killing of myeloma cells by natural killer cells when you reduce the expression of these Siglec receptors. So again, it's a little bit like, if you were to use, a PD-1 inhibitor, targeting PD-1 on a T cell, or if you were to use a PD-L1 antibody or inhibitor, targeting PD-L1 on the tumor cell, that you'd expect to see an enhanced effect, and this is what we've seen.
Mm-hmm. So a couple questions. First, how do you measure for this the thing you called ST3-GAL6? How do you, because you said in different patients you saw different levels of this.
Yes.
And might that be an indication for a patient to go, oh, this is why I'm not responding to my drugs, or why I'm becoming resistant to my drugs. I have too much of this, and that's why.
Well, I think
my first question.
While of course I have a particular interest in this particular gene. This is one of many genes that could be playing a role in any individual patient. So I wouldn't want to overstate the importance of this gene in isolation, but certainly one way of assessing the level of expression of this gene would be by doing gene expression profiling. Another way is to look at the levels of RNA by a technique known as RNA sequencing. Now, in an individual patient, the value of looking at this it's difficult. I think things like gene expression are probably more applicable to, if you like, a population or the whole group of patients, and looking at a level among a large group of patients, rather than and seeing the significance of high versus low, rather than just trying to personalize this. I think we're some way away from using these technologies on an individual patient basis. But what I can tell you is that the expression of the ST3-GAL6 is more important really for the generation of the sialic acid that would bind to selectins rather than to Siglecs. Now, selectins are these are proteins that are on the walls of blood vessels, always expressed on the walls of blood vessels or endothelial cells in the bone marrow, but are also expressed in blood vessels in inflamed tissues. Now, myeloma cells express a type of sialic acid linked to another sugar called fucose, and this combination gives rise to something that we call a selectin ligand rather. So it's a sugar that will stick or bind to E-selectin, which is on the walls of the blood vessels. And basically, what this interaction serves to do is to slow down blood cells as they're traveling through the circulation, or indeed tumor cells. And in fact, in the small little blood vessels, blood cells are traveling along at a couple of 100 miles an hour, literally. So if a blood cell is to get out of the circulation, for example, into the bone marrow, it has to slow down, stop, and then exit. And selectins, by binding to these selectin ligands on the surface of cells, allow the cells to slow down and stop. And we think that E-selectin plays an important role in how the myeloma cell exits the circulation and gets into the bone marrow. So it's functioning as a gateway into the bone marrow. And some years ago, I think we would have thought that myeloma is just little pockets of disease that stay resident in different sites in the bone marrow. But the idea that cells would leave the bone marrow, go into the blood, circulate, and end up in another site of the bone marrow wasn't something that people, really, considered. But work by people like Irene Gabriel has shown that, in fact, circulation from one bone marrow site to another is actually a very important feature of myeloma, and we think that these E-selectin ligands play an important role in that circulation and certainly in how myeloma cells get back into the bone marrow. And so this ST3-Gal6, by high levels of this, will lead to, we think, higher levels probably of these E-selectin ligands that participate in this process. And once cells are then in the bone marrow and perhaps in contact as well with E-selectin, they can be more resistant to chemotherapy. So this is a drug resistance behavior that is actually quite separate from the immune resistance. So I've been collaborating with a company called Glycomimetics, a small Maryland-based company, who, as the name implies, they are in the business of developing small molecule mimics of sugars. And what they have actually developed is a small molecule that is sort of a mimic of sialic acid and fucose that basically binds to or blocks rather the binding to E-selectin. So it's an E-selectin inhibitor. And working with this company, what we were able to show is that when you take myeloma cell lines, and if you look at cell lines that we study in the laboratory, they don't actually express a very high level of these particular selectin ligands, the E-selectin ligands. But it's possible to purify the cells that are positive for these particular sugars. And when you put those myeloma cells into mice, very interestingly, the myeloma is completely resistant to bortezomib. Now,
wow.
if then you add in this E-selectin inhibitor, basically you reverse the resistance. The myeloma is now sensitive again to the bortezomib. So we believe that in this case that these particular E-selectin ligands on the surface of myeloma cells may play a role. I'm not saying it'd be a very big leap to say that this is the reason for bortezomib resistance, but may play a role in some cases in the resistance to bortezomib. So if myeloma cells have lots of these sugars, maybe they're more likely to stay in the bone marrow where they're more resistant to chemotherapy. But if we can block that sort of gateway into the bone marrow when the cells are circulating and more of them accumulate outside the bone marrow, then they're more vulnerable to treatments like bortezomib. So
you can kill them easier.
So you mentioned the experience briefly in AML, and by getting involved with the company, a few years ago, they invited me, because I have an interest in leukemia as well, to get involved in their first clinical trial in AML. And so we were lucky enough to treat the very first patient ever treated with an E-selectin inhibitor here in Ireland for the treatment of AML. And that drug basically has shown sufficiently promising activity in AML, and in AML it's thought again that expression of these sugars, selectin ligands on the surface of the AML cells that this binding to E-selectin is mediating resistance. So there's very good data in AML now that it's safe to give this drug in combination with standard intensive chemotherapy, and that this increases the response rate over and above what would have been expected in relapsed AML patients. And in fact, it's extremely well tolerated as well, to the extent that this compound in some way seems to actually provide some protection against the development of a complication known as mucositis, which is inflammation of the gut. And anybody who's had a stem cell transplant for multiple myeloma is probably familiar with mucositis,
right,
because you probably had it. So it turns out that mucositis, part of the problem is that you get inflammation in the gut from the chemotherapy, and one of the mediators of inflammation are macrophages, and macrophages actually migrate to the site of inflammation. And as I mentioned earlier, that blood vessels in inflamed tissues increase the expression of E-selectin, so you get these inflammatory cells moving to the location of inflammation, and this can be stopped by using this small molecule inhibitor. So in a way, it's the best of both worlds, that you're able to enhance the effect of the chemotherapy while at the same time mitigating to some extent the side effects of the chemotherapy. So as I said, based on the promising data in AML, the drug now has breakthrough designation in relapsed and refractory AML, and we've recently initiated a trial with GMI 1271 in multiple myeloma with glycomimetics, and well, I should say they have initiated a trial, but I'm leading the study. And it's very, very early days yet. We've only enrolled a few patients so far, but other sites are coming on board outside of Ireland, in Germany, in the UK, and in Denmark. And hopefully, before long, we'll have greater patient numbers and some data to share.
So for that study, how is the study created? You're using this for this E-selectin inhibitor for relapse, I'm assuming, like relapsed refractory myeloma patients. Are they getting any other treatments
actually,
with it, or?
No, actually, at the moment, I think which is a great thing, because there's a wealth of different agents available now to test in patients with relapsed multiple myeloma, and more and more promising things becoming available by the day, that how to fit in an agent like this is a challenge. So one of the things I would point out is that this drug on its own would not be expected to have any activity in multiple myeloma. What it's doing effectively really is, if you like, blocking the stickiness of the cells to the blood vessels, and it's sensitizing the cells to treatment, but it's not actually killing any cells on its own. So what we decided to do was to do a study looking at this in patients who had less aggressive relapse. So patients who were in relapse, so there's basically two categories. Patients who are in relapse who were responding slowly to a proteasome inhibitor, so less than a VGPR after 3 cycles of treatment. Now that could be with
that's like a partial response, right? If people don't know what VGPR is, yeah.
So VGPR, so a very good partial response. So anything less than a very good partial response within 3 cycles of proteasome inhibitor-based therapy. So it could be bortezomib or Velcade, it could be, Kyprolis or carfilzomib. And those patients then are, considered, eligible or potentially eligible, providing they fulfill other sort of standard criteria. So that's one patient population. And then the second patient population are patients who are slowly biochemically relapsing. So somebody maybe who's been on Velcade, who has achieved a response, but is now slowly starting to lose that response, but has no end organ damage, just that their M protein is starting to slip a little bit. Those are the categories of patients, and the intention is simply to add in the investigational agent on top of the existing treatment that the patient is already getting. So therefore,
that makes sense.
If a patient is slowly responding, is sort of like, in a way, hit a wall, that their M protein just isn't dropping any further, and now we add in this drug, and with continued therapy, their M protein then starts dropping. Well, it's not definite, but there's a stronger argument that the only change really was the introduction of the new inhibitor. And the same, if we see somebody who's biochemically relapsing, and then that their M protein sort of turns the corner and starts coming back down again, the only thing we've done different is to add in the new E-selectin inhibitor. So, it's a challenging study. I kind of won't dispute that. It's going to take, I think, a while to, build up, experience with this. And, we with our first few patients, we've also started with a somewhat lower dose than was eventually found to be, the best dose in AML. So it's a phase 1 study, so it's going to take a little bit of time to learn how best to use this drug. It's also, at the present time, it's an intravenous infusion, so it takes, about 20 or 30 minutes to give this as a short infusion, and it's delivered the day of and the day after, treatment. So it could be more convenient. I think the development of, for example, a subcutaneous formulation, and of course the Holy Grail would be an oral drug. But I think for something like this that works in this way, that would probably be challenging. So I think probably the best that we're likely to get to is, in the short term at least, is a subcutaneous formulation. But it's likely that this drug will be explored in other settings in myeloma. But we have to kind of go with the data, the preclinical data that we have for the time being, and the data that we have, most strongly, justifies, trying to reverse, resistance to proteasome inhibitors.
Mm-hmm. Well, it sounds really exciting. So it sounds like you're working on 2 different but kind of complementary approaches. So getting rid of the sialic acid on the cells so that NK cells can do their work, and then this E-selectin inhibitors that basically clear the path so the blood can keep flowing and they don't have a chance to escape into the bone marrow. I have a prior question about the sialic acid. So you said when you basically clear it off, then the NK cells can kind of do their work. So if you wipe out this sialic acid on all cells, is there any downside to doing that?
Yeah,
and then is there anything in clinical trials that is happening on that right now?
Yeah, so that's a very, very good question. And I have been, collaborating with investigators from Scripps in San Diego who developed a sialyl transferase inhibitor. So, a drug that actually blocks the enzyme activity of the different sialic acid enzymes. And if you give this drug in high doses to mice, it's very, very effective at removing sialic acid from cells. And sialic acid is present on normal B cells, T cells, but it's also present on cells in for example, in the brain, in the kidney, in the liver. And the single most important toxic side effect when this drug was delivered to mice was a reduction of sialylation in the kidney, and this led to a major leak of protein, inflammation and a protein leak, something that we call nephrotic or nephritic syndrome. So basically it caused fatal kidney damage to the mice in very high doses. Now, when you give smaller doses, and you may not need to give those really high doses. It may be possible, and it's something that we are actively exploring in animal studies. So I have a good collaborator in Maine, Michaela Reagan, who used to work with Irene Gobriel, and we're collaborating because she has a lot of experience with animal models, looking at the ability to, if you'd excuse the pun, to find a sweet spot where we can deliver just enough of the drug to get the desired effect without causing any of the the unwanted side effects. But of course, there's an entirely other approach that can be used as well, since we're dealing with a disease that involves the bone marrow. If we could selectively deliver the bone, or the drug rather, to the bone marrow and avoid exposure to the kidneys, then there shouldn't be such a problem.
Right, that would be great.
The effect does wear off, so we will get enough of an effect by delivering this to the bone marrow that potentially we could recalibrate the immune system. We could affect drug resistance, and maybe if we followed up with a bit of bortezomib, that we clear out a lot of the myeloma that is resistant because of binding to E-selectin, but also that by hopefully reactivating the immune system, we are now able to engage the natural killer cells and maybe macrophages as well, and we think that this sialic phenomenon is important on macrophages as well. So we're exploring ways, and in the university here in Galway, we have a large biomaterials group, and I'm actively collaborating with the director of a very large network called CURAM, which is the Irish for cure, which is all about biomaterials. So Professor Abe Pandit and myself are trying to develop a nanoparticle delivery strategy that could selectively deliver a payload of a sialyltransferase inhibitor into the bone marrow as one possible approach. Another approach, though, that I alluded to, that Carolyn Bertozzi is pursuing in Stanford, is to use antibodies as a means of delivering an enzyme like sialidase. And you could imagine, we have a number of antibodies that work quite well in myeloma as it is, but you could imagine that maybe you could potentiate the effect of those antibodies even more by adding some sialidase or something to them and targeting the myeloma cells directly.
Yeah, then they'd be more targeted. So one question that we haven't talked about that I know probably everyone thought about when they heard that we were doing a show on sugar molecules is a patient's diet relevant at all? If someone, you've heard, a lot of us have heard, well, sugar feeds cancer, and maybe that's why there are more sugar molecules on top of these cells. Do you want to discuss that? Because I think that's something really important for patients to understand.
Yeah, I think as I said at the outset, that there are different types of sugars, and the sugars are a very large family. And yes, sialic acid is a type of sugar, but what a sugar really is, it's a sort of a compound that's made up of in a particular structure of carbon, hydrogen, and oxygen, molecules. And there's many different types of sugars. Then we have what we call monosaccharides, polysaccharides, and so on. And glucose is a very specific, fructose. People have heard of high fructose corn syrup and so on. These are very, very, they're high energy types of glucose, or of sugar rather, that are specifically there to kind of for the purposes of energy. And this is obviously too much of these in the diet, is what's linked to obesity and everything. But sialic acid isn't really linked to diet, as far as I'm aware anyway. But in terms though of certainly the link between obesity and immunity and cancer, I did that time we met in Madrid. I'd actually attended a session there where I heard quite an interesting talk from a lady from MD Anderson, and she was talking about how obesity and kind of a high sort of fat and high sugar diet in mice can lead to changes in the immune system. And one of the things that she discussed was the development of an increase in myeloid-derived suppressor cells. Now, there's a lot of literature, and there's certainly increasing literature about myeloid-derived suppressor cells in multiple myeloma, and they're one of the bad cells that's in the microenvironment in the bone marrow in myeloma. And these myeloid-derived suppressor cells, they express PD ligands. They also encourage the development of regulatory T cells, which are the type of T cells you don't want because they're dampening down your immune response. They're reducing the number of your sort of active cytotoxic or killer T cells, and they're also actually dampening the activity of the NK cells. So in that sort of way, not related to sialic acid, but certainly obesity and too much sugar and everything can have very detrimental effects on the immune system that can ultimately promote cancer.
So I guess that's the key point, is regardless of whether it causes more sialic acid or not, it's still important to just be healthy and to eat a low-fat, healthy diet and probably exercise.
Yeah.
Yeah.
Couldn't agree more.
So interesting. Yeah, so very interesting. Well, I want to open it up for the opportunity for people to ask questions of you. So if you have a question for Dr. O'Dwyer, you can call 347-637-2631. And then I have one follow-up question. If everyone's shy. We have a lot of callers on the line, so just press 1 on your keypad if you would like to ask him a question. And then in the meantime, while we're waiting, I would like to ask about, I saw in your research that there was some kind of NCI match signal finding trial. And I just wanted to ask you about that because it looks really, really interesting.
Well, I'm not sure where you saw that, but I'm not personally involved in that. But I do know what it is. It's where, across a number of different, cancer types that, if people aren't responding to standard treatments, but they're found to have, a specific mutation or, that usually a mutation that might be, responsive to a number of different investigational or other agents that are out there, then patients can be enrolled on this study. So there's a large panel of different kinase inhibitors, for example, targeting things like BRAF. There's a kinase called ALK. Various different things that aren't necessarily expressed in just one cancer, that can be expressed across a range of different cancers. So, for example, everybody's very familiar, I think, with BRAF as being an important target for malignant melanoma, and people sometimes confuse myeloma with melanoma, but in fact, a small proportion, maybe 5% of patients with multiple myeloma, especially in the high risk kind of relapsed phase, and particularly enriched, I think, in patients with extramedullary disease, can have a BRAF mutation. So it's not something that's exclusive to melanoma. So if, for example, somebody happened to have some sequencing done or had a mutation found, and there was a drug on this list that could be useful to them, then they could potentially get on that NCI match trial.
Okay.
I believe that myeloma is one of the diseases that is on the list.
Okay. Well, I have one final question. I think everyone's being very shy. So I have one final question for you. When you talked about the technology to kind of clear up the sialic acid and how you're testing it in lower doses and things, where does that stand in terms of clinical trials, and when do you anticipate that coming to clinical trials?
I think we're a long way, to be honest, from that. I think if we can, show in the research that we're doing in animal studies that this is something that really does work, then what we would need to do is, potentially, I suppose, patent this or license this technology to, a pharmaceutical company, who have the deep pockets to actually, do the necessary work that would get this all the way to a clinical trial. So I think we're really exploring at this stage more, the biology and understanding, the importance of sialic acid on myeloma, but we're a long way, I think, from actually translating this into a clinical trial. I think, though, that there are other approaches, and I mentioned, the possibility of instead of removing the sialic acid from the surface of the cells, if this turns out to be important in myeloma, and of course I emphasize we've only done work in the test tube, if you like, in the laboratory, but if it turned out that this was really important, we can actually knock out the receptors on the NK cells, and
interesting.
And that's something that
do it backwards, kind of
done in a much faster way, and could get actually to the clinic much faster.
That's really fascinating that you could go from the immune system side and not from the sugar cell side. Interesting. Wow. Well, it's what you're working on is just so fascinating. It sounds like the E-selectin inhibitors are a little bit closer, or a lot closer, because now they're in clinical trials. But we just wanted to thank you so much for joining the show and for your participation. and explaining all of this new, because this represents really a completely new drug class for a group of drugs.
I kind of I want to thank you for the opportunity, if you like, to to share what we're doing. I do emphasize it is really relatively early compared probably to most of the speakers you have on your show. Probably their work is at a much later stage and kind of more accepted in that, and we've a lot to do, I think, to really validate this and show that this will ultimately be of value to patients. But we have to start somewhere, and I think for the time being, at least, we're on the right track.
Oh yeah, it sounds fantastic, and I think this just goes to show how complicated the whole thing is. I mean, why some people respond and why some don't, why some people become resistant, why some don't. And so what you're doing is really important.
Well, thank you.
Well, we have a caller question at 561-1149. Go ahead with your question. Hello? Oh, they dropped. Okay, caller at 847-5748. Go ahead with your question.
Yeah, Dr. I was found to have to carry the BRAF mutation, and my question is: at what step in a patient's treatment plan does it make sense, given the multitude of other available treatments? Does it make sense to try the agents that are found to stimulate for carriers of the BRAF mutations? At what point myeloma and do you think it makes sense to try those MEK inhibitors, etc.?
Yeah, I mean, I think there are people like Mark Rabb and Gareth Morgan who have personal experience of using BRAF inhibitors in this situation, and I think I have no personal experience of doing this, but I think the general concept is that just because you find a mutation in a patient doesn't necessarily mean that that's what's driving their disease. Mm-hmm. And I think the clinical context is very important. I think in a patient that's got extramedullary disease, where they have very explosive disease that's rapidly progressing, and where you find that it looks like a high proportion of the cells are BRAF mutated, then it makes perfect sense to use a BRAF inhibitor. And in that situation, clinical, very good clinical responses have been seen, but of course, because of the nature of clonal evolution and everything, it's inevitable that as you suppress that clone, but then, in time, another clone that's BRAF negative is probably going to take its place and take over again. So, the responses aren't going to be durable, but you can get a very, good response for a period of time. But if it's just there in a minority of the cells and is not what's actually driving, then, it probably doesn't make much sense. And I think, I started my career, with imatinib in what is a fairly, homogeneous disease where you've got a single kinase that if you target it sufficiently well, you can get, excellent durable responses. I think, when we all know that myeloma is so complicated, genetically, that you probably, need to be hitting multiple different things at the same time. And the more specific your treatment is, the less likely it is to be successful. So it's really combinations of things, probably, and bringing in, immunotherapy, for example, is one aspect, but it's not the sole aspect. I think the more angles that you can hit the disease from, the better.
Right. And just as a follow-up, how can one determine whether a particular mutation, such as the BRAF, is the driving mutation for a patient's myeloma?
I wouldn't have personal experience of doing this type of analysis, but I'm sure you can probably, look at, identify different clones that are present. You can probably sequence and see, what proportion of the disease that's there is actually, positive for that particular mutation. And if it looks like it's the dominant clone across, or the dominant mutation rather across all the different cells that you're looking at, well then, it makes sense to treat that. But if it's only present in a minority of the cells, probably doesn't, that there are other clones that are probably more important.
Thank you.
Okay, thank you so much for your question. Well, Dr. Dwyer, thank you so much for joining us today, and we just wish you well in your wonderful work.
Well, thank you very much. It's been a pleasure.
Oh, thank you so much. And thank you, our callers, for listening to Myeloma Crowd Radio. Tune in next time to learn more about the latest in myeloma research and what it means for you.
Okay, round two. Name something that's not boring.
A laundry? Ooh, a book club. Computer solitaire, huh?
Ah, oh, sorry, we were looking for Chumba Casino. That's right, ChumbaCasino.com has over 100 casino-style games. Join today and play for free for your chance to redeem some serious prizes. ChumbaCasino.com. No purchase necessary. Void where prohibited by law. 18+ terms and conditions apply. See website for details.